The Earth's oceans are a wonder of the natural world. They comprise the largest habitat on our planet and play a vital part in our climate system. But they are under threat. It is estimated that around 10 million metric tons of plastics find their way into our oceans each year. This is equivalent to the mass of around 65 international space stations every single day. Professor Julian Moger in the Department of Physics and Astronomy at the University of Exeter studies a kind of plastic pollution known as microplastics.
So as we know, plastics are everywhere these days. The oceans are full of plastics that have been discarded. They get washed up on beaches. You really can't get very far from finding plastic in the environment. But what we've recently discovered is that plastics break down into really small particles called microplastics. Microplastic particles are tiny particles of plastic. Micro meaning 10 to the minus 6. So 10 to the minus 6, a millionth of a meter in size, which is the same as 1,000th of a millimeter. So too small to see with the human eye, but you can see them under a microscope. We've done some work with some biologists at the University of Exeter that have discovered that these really, really small plastic particles actually pose a threat to the environment. So these particles are small enough to get into cells within biological tissues and organisms. And the main concern is that if these plastics, these microplastics, get into marine microorganisms, they can make their way into the food chain, where they can really concentrate into larger animals, larger fish, and eventually get consumed by humans. With standard light microscopy, we can visualize microplastics, but one of the key challenges for identifying these microplastics in the environment and in marine organisms is that with conventional light microscopy, you can't tell the difference between tiny particles of food and tiny particles of man-made plastics. And that's where we applied physics to shed some new light on this global challenge. In this actual lab, this is where we did the work. And what we're doing here, we're using very, very short laser pulses down to 100 femtoseconds in duration. So 100 times 10 to the minus 15 seconds in duration, which is extremely short. The advantage to doing this is we can hit extremely high peak powers. So for an example, 1 milliwatt of laser power, which is equivalent to the sort of power that you might have in an eye-safe laser pointer used in a lecture or a classroom. With that level of power, we can hit about a kilowatt of peak power. And by doing that, we can excite vibrational resonances within molecules that allow us to extract information about what they're made of. And this is exactly what we did with the microplastics. We were able to tune into the molecular vibrations of the plastic. So one of the advantages of these non-linear imaging methods that we can excite with these short pulses is that we can overcome scattering. So if you want to image inside biological tissues, it turns out that light scattering is the main thing that stops you from seeing deep inside tissues. So you'll all be familiar with the reason why the sky is blue and at night time the sunset is red. And it's exactly the same phenomena that we use here to see deeper inside biological tissues. So Rayleigh scattering is the phenomenon that governs light scattering. So Rayleigh scattering, the amount of scattering is proportional to 1 over the wavelength to the power of 4. So that's the reason that the sky is blue, because the white light from the sunshine hits our atmosphere and the blue light gets scattered far more than the red light. So everywhere we look in the sky, we see blue light because it's scattered far more than the red light. And then at sunset, when the sun is lower down in the sky and we're looking directly at the sun, the longer wavelength red light is traveling straight towards us, and all the other colors of light have been scattered away, and that's why the sunset is red. So we make use of that phenomenon in nonlinear optical imaging because we're working beyond red wavelengths. We're working in the near infrared at maybe twice the wavelength of red light, and because Rayleigh scattering is proportional to 1/4 power, it makes a significant difference in how far we can see into the tissue, and in some cases we can see up to several millimeters into tissue that would otherwise be scattering with visible light. And what we were able to see, we could actually confirm for the first time that microplastics do indeed accumulate inside marine organisms. We detected microplastics inside zooplankton, inside crab gills, inside fish gills, and this provided evidence to suggest or to confirm that microplastics do get retained inside marine organisms and can make their way into the food chain. Until recently, the manufacturers of cosmetic products such as soaps, face washes, shampoos were actually putting microplastic particles into their products to make them work better. And it turned out that around 4,000 tons a year from the UK alone were finding their way into the ocean in microplastics purely through these cosmetic products containing microplastics.
Thanks to Professor Moger's contribution, by 2021, the governments of 15 major countries, including the UK, the US, and some European countries, had banned the use of microplastics in cosmetics.
This was one of the most exciting pieces of research that I've done because it was an opportunity to apply some cutting-edge physics and laser technology to something that's actually had a tangible outcome on society and the environment, which is something that I'm really proud of.